learn about Western blotting. Firstly, my name is Siti Nur Syuhada binti Muhammad Zukri. My group members Nur Arisa Husna binti Muhammad Yusri, Sharifah Nur Shakira, Azahra binti Syed Zulkarnain, Fisha Nur Ilana binti Da'in Idris, and Farihatul Umaira binti Ahmad Tajuddin. So, what is a Western blotting? Western blotting is a scientific method for detecting a particular protein in the blood or tissue samples. The procedure includes separating the proteins in the sample using gel electrophoresis, which sort the proteins based on their size. The separated proteins are transported from gel to the surface of membrane. The membrane then is exposed to the antibody that is specific to target protein. The binding of antibody is measured by radioactive or chemical type. Western blots are occasionally used to diagnose illness such as confirming infection, like HIV, or identifying markers for certain genetics or autoimmune disease. The principle is simple. Proteins are first separated based on their size using STS page, which is a type of gel electrophoresis. After separation, the proteins are transferred from the gel onto the membrane, usually nitrocellulose or PVDF, through a process called blotting. Next, the membrane is blocked to prevent non-specific binding, then it's incubated with a primary antibody that binds specifically to the target protein. After that, a secondary antibody is added, which is bind to the primary antibody and linked to the enzyme or fluorescent tag for detection. So, step 1 is a virus isolation. We break open the virus to release its protein using a tube and centrifuge. In step 2, we get a protein solution which includes the protein we want to detect. So, step 3 is a STS page. Here, we separate the proteins by size using a gel. Smaller proteins will move faster. In step 4, we transfer the protein from the gel onto the PVDF membrane using electric current. Step 5 is antibody probing. A primary antibody sticks to the target protein, then secondary antibody with enzyme attached to the first. Step 6 is detection. The enzymes give off light, and then we use an imaging machine to see the protein as a visible band. So overall, Western blotting helps us to find a specific protein using biology, chemistry, and also electrical tools.
Hi, my name is Arisa Husna, so I will explain you about the sample preparation. Firstly, lyse the cell or tissue. By lyse the sample by using the lysis buffer RIP, which is the command buffer for obtaining maximum protein yield. Then, add the protease inhibitor and mix them by piping up and down. Next, incubate the lysis buffer sample on ice for 30 minutes and centrifuge the sample for 10 to 15 minutes at a 4 degrees Celsius. Then, collect and transfer the supernatant, which is containing protein, into a new tube. Next, we are going to determine protein concentration by testing a small portion of lysate with an available protein quantitation assay such as BCA assay. This will assist you in loading equal amount of protein into your gel. Next is prepare the sample with a loading buffer. To reduce and nature the sample, by dilute the sample with a loading buffer such as traditional laminate buffer. Then rotate the sample and incubate at 95 until 100 degrees Celsius for 5 to 10 minutes to completely denature the protein. Next is protein electrophoresis SDS page to load the gel. After assembling the gel apparatus, place the SDS page gel into the casing. Fill the inner and outer chamber with a running buffer until the gel well are completely covered. Then load the protein ladder as a molecular weight at the first link and as a meter protein separation. Load the sample by using a micropipette to insert the tube very close to the well and expel the sample. Next, we are going to run the gel, close the electrophoresis unit and connect the power supply. Set the voltage at 180 volts and run it for approximately 1 hour or until separation of sample touch the bottom of gel. After that, protein separation disassemble the gel. Remove the gel from the casing and cut the top of the gel. Float the gel into the container that contains buffer carefully. Shake on the shaker for 5 minutes and then the sample ready for the next step.
Hi and Assalamualaikum. I'm Farihatul. Today I will explain about the techniques used in Western blotting after protein separation. First, protein transfer. The protein are transferred from gel onto a membrane like nitrocellulose or PVDF to allow antibody detection. This step relies on sandwich technique. The gel and membrane are carefully assembled between buffer soap, filter paper and sponges. The typical sandwich order are sponge, filter paper, gel, membrane, filter paper and another sponge. This setup will make sure everything is in place maintain moisture and pressure to help protein transfer properly when the electric current is applied. Remember, it's important to remove air bubble so that the transfer is smooth and even. There are 3 ways to do the protein transfer. First, wet transfer. That takes 1 to 2 hours to work. It works best for large protein because it involves in submerging the sandwich in buffer tank. Next, semi-dry transfer, which is faster than the wet transfer, around 15 to 45 minutes and less buffer is used. Last but not least, dry transfer. It is the fastest method using special tools and doesn't require any buffer soaking. After protein transfer, a blocking solution like non-fat dry milk or BSA is used to stop the antibody from sticking everywhere. Then, a primary antibody will be added to find the target protein. Next, a secondary antibody is added to bind with the first one. This second antibody has a marker like enzyme or fluorophore for the detection. Finally, the protein bands can be seen by using methods like chemiluminescence, colorimetrics, or fluorescence. The darker the band, the more protein is present. So overall, the sandwich technique, blocking, antibody steps, and detection have helped us specifically identifying the protein clearly in Western blotting. Thank you.
Now let's talk about the principle. The principle of Western blotting is the interaction between the proteins and probes used for the detection of the target proteins. Firstly, to obtain the proteins on the geometry for Western blotting, they are separated via SDS page based on their molecular weight from its biological samples. Then, those proteins were transferred onto a membrane such as nitrocellulose or PVDF membrane where they are immobilized. The transfer of the protein is known as blotting. However, to prevent non-specific binding of antibodies, the membrane is treated with a blocking solution to reduce background signals. Common blocking agent used is bovine serum albumins. Next, the protein on the membrane can be detected either by a labeled primary antibodies that bind directly to the protein or by a labeled secondary antibodies that binds to the primary antibodies. Hence, to avoid false result, both antibodies need to be specific to their targets. After that, the target protein is visualized by introducing substrates for enzyme linked to the secondary antibodies or by using fluorescent detector. Therefore, a signal is generated that indicates the quantity and presence of the target proteins. The probe on the antibody is often an enzyme such as horseradish peroxidase or fluorophore that produce color or light when it reacts with a specific substrate at the antigen antibody binding sites as shown in the diagrams. As a result, to verify the protein's identifications and validate the experiment, bases are compared to positive controls, negative controls, and molecular weight markers. Positive control confirms the protein is detected correctly. Negative controls checks for non-specific binding. Lastly, to evaluate particular protein expressions, change, or interaction within the sample, the Western blotting data is analyzed. Therefore, densitometry software can be used to measure band intensity, yielding semi-quantitative analysis on protein expressions levels, which is widely used in medical field for diagnostics.
Next, I will explain the scientific and diagnostic application of Western blotting. Let's begin with the scientific application. Western blotting is commonly used for detecting the specific proteins and semi-quantification in a simple or complex mixture of proteins. This helps to determine the relative amount of protein in different samples. Next, it is used for post-translation modification studies such as the alterations, irreversible phosphorylation states of various proteins, which help in understanding the signaling pathways. Now we continue with the diagnostic application of Western blotting. This method supports the diagnosis of infection disease such as HIV. Based on the CDC guidelines, a positive HIV Western blot must have at least 2 of this band. If not, the result is considered negative. It also supports the diagnosis of non-infectious disease where it detects disease-related autoantibodies and protein isoforms that are common in autoimmune disease, cancers, and prion disease. In conclusion, Western blot is a crucial technique in both research and clinical setting. It is widely used for protein detection, semi-quantification, and antibody analysis. Although it has some limitation in sensitivity and performance, ongoing enhancement in tools and technology will help overcome this challenge and improve its reliability in the future.